Okay, so let's review the immune system, which is chapter 43, the immune system. And the first section was really all about innate immunity. And if you remember, innate is immunity that you're born with. So 43.1 was innate immunity you're born with. So you don't acquire it. Remember, that's the second kind, is acquired. So innate immunity. So you have the first thing is your external defenses. So can you remember off the top of your head what the external defenses are? They are your skin. Remember, your skin is a barrier that keeps things out. And we have mucous membranes. So remember, we talked about lining the cells of your trachea, in your respiratory system, in your nose, and then there are secretions, sweat, tears, all right, are examples of secretion. Then, so that was your external, but remember that a lot of things get by that, and so then we also have internal defenses that are inborn, that you're born with, and are non-specific. I should put in non-specific, meaning that they act the same no matter what, if it's any kind of virus, any kind of bacteria, the same thing happens. All right, so your internal defenses, remember you have your first thing that you have is your phagocytic cells. So remember that they take in things through endocytosis, and phagocytosis, remember, is a type of endocytosis. And then you get your microbe in there, and remember your lysosome contains, what does it contain? Enzymes that goes and fuses with it and digests the pathogen. So an example are macrophages and dendritic cells. Remember, these guys can be found in your lymphatic system. So macrophages, your lymphatic system. So you want to review your lymphatic system and lymphatic vessels and lymph nodes, and so on. So that's your macrophages. The second thing is that second kind of internal defense can be the natural killer cells. These guys kill infected cells. And this is non-specific, so it kills any type of infected cell. Then, let's move down here. The third thing is antimicrobial proteins. And we talked about the complement system. And interferon. So what do these do? The complement system will cause lysis. And the pathogen. So this is, look at figure 4319. that illustrates that and how that happens. And interferon, remember, is released by virus infected cells and protects non-infected cells. So read up on that and how it does that. So those are your antimicrobial proteins. And then we can add here the fourth thing of internal defenses is the inflammatory response. And so what figure was that? Let's see here. The inflammatory response is figure 43.6. So relook at that. Let me show you what it looks like here. It's this figure right here, 43.6. So read up on that, the role of histamine, and how the blood vessels dilate, become leaky, the clotting elements go, and the phagocytic cells go from the blood to be able to take in the pathogen through phagocytosis. So be familiar with the inflammatory response. And so that is also, all of those are non-specific. So all of that is the innate immunity. So the second section, 43.2, let me turn my page over here, is all about acquired immunity. So 43.2 is acquired immunity. This is immunity that you get or acquire during your lifetime. So you are not born with it. So, so let's go over that there are two different types of cells. Remember, there are the cells involved in main cells are lymphocytes. And there are two kinds. Remember, there were B lymphocytes. We call those B cells. And we have T lymphocytes. We call those T cells. And remember that there were two kinds of T cells. There's helper T cells, and there are cytotoxic T cells. All right, cells up here. And remember that these guys are going to fight an infection against a pathogen. And pathogens, remember, these are bacteria or viruses, and what is it on the surface that we actually recognize as being foreign? If you remember that a bacteria, if this is a bacteria, the bacteria on the surface of the cell membrane has an antigen. So when we look at this, let me see, I didn't bring my colored pens home. This whole thing is, remember, an antigen. And each one of these is an epitope. Got my colored pencils there. And it's the shapes on these epitopes that the B and the T lymphocytes are going to react with and respond to. And that's how they know that this bacteria is not part of yourself. So B cells have an antigen receptors. So if we look at the structure of a B cell, the antigen receptors on a B cell are Y-shaped. So on a B cell, they look like this, and notice that, and there's lots of these, you can have 100,000 of them on the one cell, but this fits into the shape here of this epitope. So therefore they have a matching shape, and all the shapes on this particular B cell would be the same. Like that. And there would be lots of them. So this is your B cell. And so this is these are called your antigen receptors. And then a T cell. So let's look at the T cell here. The T cells are a little bit different in that they also have a receptor. And it doesn't matter, the receptors for the helper T cell and the cytotoxic T cell look the same. They're just like this. So notice the B cell has the Y-shaped and this one has like this. All right, so this is also a receptor. The thing is that the only thing different is what they're actually going to bind to. So a B cell actually binds to the pathogen. So right here, this actually binds to the antigen on the pathogen. And that is figure 43.17. So if we look here, (43:17) is right here. So the B cell, here's that little figure here, the antigen receptor actually binds to the pathogen itself. Versus your T cells, whether or not it's helper or cytotoxic T cells, they're binding to antigen fragments. So a helper T cell, the antigen receptor binds to an antigen fragment. And the fragment is displayed by an antigen presenting cell. And APC is the antigen presenting cell. The cytotoxic T cells binds to an antigen fragment displayed by an infected cell. And so that's what we see here. This is 43.9 or 19. No, 9. Oops, I had it right the first time. Figure 43.9. So if we look at this here, notice your cytotoxic T cell and your helper T cells have that one little display. And here's, if we see here, the antigen presenting cell engulfed the microbes, displays the antigen, and that helper T cell binds to that. Here's an infected cell, displays the antigen, cytotoxic T cell binds to that. One other thing about the B and the T cells are where they are made. So these guys, B cells, are made from stem cells, and then they finish development in, or differentiation in, the bone marrow. So review that. And these guys are made from stem cells. You might want to write made from stem cells. And they finish in the bone marrow. These guys finish development. Didn't leave enough room here. Finish development in the the thymus. Okay? So remember that when you're making B cells or any type of lymphocytes, T cells, and so on, they are developed in and go through differentiation and division. So let's talk about the clonal selection of lymphocytes. So an example is with the B cells. So remember that the clonal selection is that you have many different types of B cells. So they have many different antigens. Shape, and the difference is that they're the shape of the antigen. So this one, antigen receptors, I should say. So this is like half circle here, and we can do shapes like that. So with 3 different B cells. So you have lots of different B cells, and remember your antigen molecule is what you're reacting with. So if this is your antigen, it only will bind to one type of, in this case, B cell. And so this right here, let's say, oops, let's make it a different shape here. Let's make it triangle here. And this will only fit into this particular B cell. So this one's is the one that it's going to bind to, and that's going to cause this to divide. So it's going to, so this is the selection. This is why it's called clonal selection. It's going to divide and make many B cells that have those same antigen receptors. So they divide. Like this and differentiate. So this is the dividing part. And then they differentiate into 2 different types. And so the clone, they differentiate into memory cells. They look exactly like this. And so these guys are long-term. Remember, they stay around a long time. They're long-lived. And then that makes the plasma cells. They differentiate. So this is the differentiate part. Plasma cells, and these guys do not have the antigen receptors, and these guys secrete what? They secrete antibodies that look just like the receptors. Okay, so they're not secreting the receptors, they're making separate antibodies that secrete, that are secreted. And that's your clonal selection. And I use the example that this happens with B cells, but this also happens with helper and my pen is running out. Let me switch pens. Helper and cytotoxic T cells. Okay, so it happens with all of them. The dividing, the activation, the dividing, and memory cells and active cells. So remember, the antibodies are important. Remember why they're important on this figure here. 4319, that they neutralize the viruses or bacteria by binding to them so that they can't infect other cells. They make them kind of stick together so that they're easily, in these cases here, easily taken out by the macrophage. And so that's what, and they can, if we look here, the macrophage here is engulfing that antibody surrounded bacteria, and the antibodies help aid that by the binding of them to the macrophage and engulfs it through phagocytosis and digests it. So antibodies are important to, remember, promote phagocytosis. Okay. So these guys, remember, help stimulate phagocytosis by those phagocytic cells. Okay. So remember the memory cells, the long-term stay in your body in the long run and make the second response happen a lot quicker. And so so that's what I want to talk about next is primary versus secondary immune response. So review that. Remember that this is figure 4313, primary versus secondary immune response. And so that the second time you come into contact with something with the same antigen, you have a much faster fight against that infection. Okay, so this is the figure I was trying to get to you to be able to see. So this is 4313. So remember that the first time you come into contact with the antigen, it takes a while to even begin to make the antibodies, and that's because you have to go through that clonal selection process. So the very first time you have to pick which B cell is going to divide and differentiate by which one binds to the antigen. Then it divides and differentiates into memory in the plasma cells, which secrete the antibodies, and that's why you see the antibodies going up. And then they go back down, and help to fight off the infection with stimulating phagocytosis, and the virus or bacteria is gone. So then the second time you come into contact with that same antigen on the same pathogen, you get a much quicker response. Why? Because you already have those memory cells, and so they can begin to make antibodies immediately. You don't have to go through that differentiation process, or the dividing and differentiating process, and the memory cells can quickly make the antibodies and therefore fight the infection off much quicker. And you have to do the same thing to another antigen. So that antigen is in those memory cells that were created from the first exposure with pathogen A is not going to have any effect on pathogen B. So you get a new pathogen, you have the same first primary immune response where you have to develop those memory cells. Okay. So that is your primary and secondary immune response. All right, so then 43.3 was all about the difference between humoral and cell-mediated immunity. All right, so this is where we did that big concept map. And so really, I feel like the best way to review that is to redo that concept map. So actually, I started it going sideways here, but for this, I'm going to go ahead and go like this and just put it underneath here like this. So if you remember, immunity divides into innate, which we already talked about, and acquired, right? And innate, we had, remember, our external and our internal. defenses. And remember, part of those internal defenses was the phagocytic cells, like the dendritic cells. And so remember that we said that once they engulf that pathogen through phagocytosis, there's the pathogen in there. And so then you have the vacuole with the pathogen, and remember the lysosome fuses with it, digests the pathogen, but remember that that cell presents the antigen. So that's called an antigen presenting cell, or APC. So it presents that antigen. And so this is an antigen presenting cell, like dendritic cells, macrophages. And remember that that's going to stimulate your helper T cells. So with acquired, remember you have humoral immunity, and this is with your B cells. And you have cell-mediated with your T cells. And remember you had helper T cells in the center that wasn't really part of the humoral or the cell-mediated. But what they do, remember, they're stimulated by the antigen-presenting cell. So you have an antigen-presenting cell presenting the antigen. So this is your APC. Your APC releases cytokines. So these guys here are cytokines. And you remember what the cytokines do? The cytokines are going to stimulate the helper T cell, which helper T cell is going to be activated is the helper T cell that has the antigen presenting, or antigen receptor, that has the appropriate shape. So this is your helper T cell right here, helper T cell. And remember that it has a receptor in the membrane that these cytokines can bind to. So the cytokine binds to that, causes triggers a signal transduction pathway, which then leads to a response. And that response is a couple of things. The response causes it to release its own cytokines. So it releases its own cytokines, which then communicates with itself, which then triggers it to divide and differentiate into memory and active helper T cells. And memory helper T cells. And these active helper T cells, then, they're going to divide and differentiate and are going to be active. So they are going to be releasing cytokines and triggering themselves like this to divide some more. But also remember the cytokines also stimulate the cytotoxic T cells. So your cell-mediated version is with your cytotoxic T cells, and your humoral is your B cells. And these cytokines stimulate both of those. So here's this, and I'll put here cytokines, All right, stimulate both of those. So, the helper T cells, this activation of the helper T cells are going to trigger the B cells and the cytotoxic T cells. So what a B cell does. A B cell, remember that it is a B cell. When it comes into contact with a pathogen, a B cell does, it presents the antigen on that pathogen. So let me go back to this figure here. 4317. This figure right here, 4317. So here's your macrophage, and it takes in the bacteria and presents the antigen and stimulates the helper T cell. So now you have an activated helper T cell. So that's where we're at here, active helper T cell, and it's able to release the cytokines. But what it does, notice the activated helper T cell binds to a B cell, but look at what's going on with the B cell. It has those Y-shaped antigen receptors, so they've bound to the antigen on the bacteria, and it has digested the bacteria, taken it in, and presented the antigen on the bacteria. That the bacteria had on it, and it's presenting it, and that's how the helper T cell knows what B cell to actually stimulate and communicate with it, so that it will divide and differentiate. So it won't activate any B cells that are not presenting the right antigen, and so that's how they know, all right, which B cells we need to divide and differentiate. So when we look at the B cell here, the B cell is presenting the antigen. So then this helper T cell that's active, all these active helper T cells, remember helper T cells have they have that antigen receptor site. We talked about that here in our review. All T cells have that antigen receptor. And the helper T cell with the right shape binds to the B cell. And it's active, so it's releasing cytokines. Which bind, here's another receptor, to the B cell. So cytokines bind to the B cell and trigger a signal transduction pathway, and then eventually it gets to a response. What's the response? For the B cells to divide and differentiate into memory B cells and the plasma cells. I'm going to put that these are active. So these are in your current, working with your current infection. And these guys are for longer use, remember, for long-term use for the second infection. And these guys make and secrete antibodies. Which, remember, enhances phagocytosis. So remember that we said that then overall the humoral part of the acquired immune system enhances the attack on pathogens. Enhances attack on pathogens. That's what this part does. So you're helping phagocytosis to get those pathogens out of the body. The second part of the acquired immune system is the cell-mediated part, and this is with the cytotoxic T cells. So if you remember, what figure was it? It is figure 4316. So, an activated cytotoxic T cell will bind to an infected cell. Remember, infected cells also display the antigen of the pathogen that has infected it. So that's how the cytotoxic T cells know which one cell to bind to, which cell to kill. And so remember that it releases perforin and enzymes that help to for apoptosis to happen in the target cell, or for the target cell to to go through apoptosis, it has to be stimulated, and it has to have the T cell bind to it. So it has to have the cytotoxic T cell bind to it, as well as the perforin and so on. So let's go ahead and draw that. And so your infected cell is going to be displaying the antigen. So here's your infected cell. And we know that it's infected because it has the antigen being displayed. And what's displaying it is this MHC molecule. The MHC is displaying the antigen. And when the cytotoxic T cell comes into contact with it, so remember, T cells have an antigen receptor that looks like this, and so this is the cyto T cell. So what the cytotoxic T cell does, remember, is it releases perforin as well as enzymes. So perforin, remember, pokes holes in it, and then it releases the enzymes that can go through those holes and break down and cause that enzyme triggers apoptosis. And so the infected cell goes through apoptosis. So that's what a cytotoxic T cell does. But remember that this is an active, if that's an active cytotoxic T cell, how does the cytotoxic T cell become active? It becomes active with the help of those cytokines. So the cytokines, when they actually bind to the cytotoxic T cell, triggers, so the cytokines come over here, binds to the cytotoxic T cell through a receptor, and that triggers the response, a signal transduction pathway and a response, and that response is for those cytotoxic T cells to divide and differentiate into memory cytotoxic T cells and active cytotoxic T cells. And those active ones do just what I just showed here. They go and they find infected cells and they trigger apoptosis to happen. And so we would say that the cell-mediated part attacks infected cells. So that is your acquired immunity in a nutshell, and all the different types of cells and what they do. I also would ask you to review for this section review figure 4319 and look at how antibodies work, and just make sure that you know that and review that. I also would like you to look at what MACs are and their importance. So those are the molecules that are receptors, proteins in the membrane of cells. So just review those. Another thing to review in this section is active versus passive immunization. So remember, active immunity develops naturally in response to an infection. So this is when you naturally get an infection. You go through, recognizing the pathogen, and the B cells bind to it, present it, helper T cells are activated, and this whole thing happens naturally, and you end up with memory cells and active cells to fight that current infection. So it's natural where your body is going through this acquired immunity process. So another way, it says develop naturally. An example of that is getting an infection where you actually are infected with a pathogen in your day-to-day life. But another example of that is immunizations or vaccinations. That's, remember the premise behind that you are given a weakened or dead version of the pathogen. So you don't get sick, but you go through the whole acquired immune response. So you end up making memory cells, memory B, memory T, helper and cytotoxic T, and so on, so that the next time you get infected or by that pathogen, you have a secondary immune response, and it happens much quicker. Whereas passive, this is where you're given the antibodies so you can fight the infection, but you never have an acquired immune system response because you never come into contact with the pathogen. So in what instances, when an antibody, I'm just going to put antibody, crosses the placenta from mother to baby, from mother to baby, or the antibody is given to baby through breast milk. So babies have a incomplete, very young, not very good immune system, and so mothers can give their antibodies, their actual antibodies from pathogens that they have come across, can be transferred to their baby through the placenta if they're still in the womb, or through breast milk. We can also do injections where people going into foreign countries can be have injected antibodies into them, and so they have temporary immunity because those antibodies will stay in their body for a while, and they can fight off a pathogen for a short period of time. So they never have an acquired immune system response, but they have some short-term protection because they're just given the antibodies. And the last thing for the last section is that I would like you to review blood groups A, B, O. Look at that. Rewatch the video that I talked about it in. Look at transfusions. I'd like you to review tissue and organ transplant. And then 43.5. And 43.5, I'd just like you to review allergies, and what autoimmune diseases are, and how they are affected, how they affect people, and immunodeficiency. Diseases. All right, and so that is the immune system. All right, so hopefully that gives you a good idea of what you need to know, and gives you a good overview of what you should spend time studying.